Welcome to the module on UV visible absorption spectroscopy. This technique, commonly referred to as a UV-vis spectroscopy, is essential in modern analytical chemistry and various other scientific domains. It primarily operates in the ultraviolet 200 to 400 nanometer and visible 400 to 800 nanometer regions of the electromagnetic spectrum. As discussed in our previous modules, each region of the electromagnetic spectrum interacts with matter in a distinct way. More specifically, UV visible spectroscopy measures the absorption of ultraviolet or visible light by a substance. When molecules absorb UV or visible light, their electrons get excited to higher energy molecular orbitals, transitioning from a ground state to an excited state. The specific wavelengths of light or energies of photons absorbed by a molecule are indicative of its molecular structure and composition. This absorption offers both qualitative and quantitative information about the substance, making UV-vis spectroscopy an invaluable tool for understanding and analyzing molecular systems. Furthermore, UV-vis spectroscopy is a good representative example of an absorption spectroscopy technique. This figure shows the primary components of a typical UV-vis spectrophotometer. First, we have the light source. Commonly, deuterium lamps are used for the ultraviolet region, while tungsten halogen lamps are preferred for the visible region. Together, they emit a broad spectrum of light, covering both the UV and visible ranges. Next, the emitted light passes through a monochromator. This component is crucial as it selects specific wavelengths band of light to pass through the sample. By doing so, it ensures that only the desired wavelength reaches the sample, allowing for precise measurements. The selected light then travels through the sample cell, where it interacts with the sample. Liquid samples are usually contained in an optical cell referred to as a cuvette. They typically hold a few milliliters of the solution and are characterized by a 10 mm optical path. The actual absorption of light by the sample occurs while it passes through the sample cell. Depending on the sample's composition and structure, it will absorb specific wavelengths of light, leading to electronic transitions. Finally, after passing through the sample, the light reaches the detector. The detector measures the intensity of the transmitted light, allowing us to determine how much light was absorbed by the sample. In UV visible absorption spectroscopy, obtaining accurate and reliable results requires the use of a reference. The reason for this is simple. When we measure the absorption of a sample, we are not just measuring the sample's interaction with light. We are also measuring any interaction that might occur due to the solvent, the cuvette, or even the instrument itself. To account for these factors, we use a reference measurement. This involves measuring the absorption of a reference, typically a solvent or a blank, under the same conditions as the sample. By comparing the two measurements, we can eliminate any background or instrumental effects, ensuring that the data we obtain is the actual absorption due to the sample alone. Using a single beam spectrophotometer, measurements are carried out sequentially. In this setup, the instrument first measures the intensity of the light source without the sample, which serves as the reference. After this, the sample is introduced and the intensity of the light passing through the sample is measured. Single beam spectrophotometers are simpler in design, but the separate measurements for the sample and the reference can introduce potential errors, especially if conditions change between two measurements. For instance, slight fluctuations in the light source intensity or drifts in the detector can affect the accuracy of the results. To prevent these issues, it's essential to ensure that both the reference and sample measurements are carried out under identical conditions and in quick succession. Despite these challenges, single beam spectrophotometers are widely used due to their simplicity, cost-effectiveness, and suitability for a range of applications, especially when high precision is not vital. The double beam spectrophotometer, whose functioning is schematically depicted in the figure, offers a more advanced approach to UV's measurements. This instrument is designed to measure both the sample and the reference simultaneously. In this setup, the light emitted from the monochromator is split into two separate beams. One beam passes through the sample cell while the other passes through the reference cell. The intensities of these beams after passing through their respective cells are measured simultaneously. This synchronized measurement ensures that any fluctuations in the light source or drifts in the detector affect both beams equally, thereby cancelling out their effects when calculating absorbance. While double beam spectrophotometers are typically more expensive than their single beam counterparts, their increased reliability and precision make them a preferred choice when high accuracy is essential. The results of any absorption spectroscopy experiment, including UVVs, are expressed using two key parameters: transmittance and absorbance. During the experiment, we measure two light intensities. The first is the intensity of the light in the reference measurement, denoted as I0. The second is the intensity of the light that passes through the sample. We denote it as I. Transmittance is defined as the fraction of incident light that passes through a sample. Mathematically, it is given by the ratio of I and I0. This can also be expressed as a percentage. Absorbance A, on the other hand, is a logarithmic representation of the fraction of light's intensity absorbed by the sample. It is calculated as the negative of the decimal logarithm of transmittance. In UV visible spectroscopy, we study how molecules and atoms absorbed light of different wavelengths. When light of a specific wavelength shines on a sample, some of it gets absorbed. This absorption occurs because the energy of the light can excite electrons in the sample from a lower energy level to a higher one. By varying the wavelengths of the incident light and measuring the intensity of the transmitted light, we can generate an absorbance spectrum. This spectrum typically plots the absorbance of light against the wavelengths. For atoms, these absorptions result in sharp, narrow bands corresponding to transitions between electronic levels only. However, molecules have additional vibrational and rotational energy levels superimposed on their electronic energy levels. This means that a molecule can absorb light at multiple wavelengths, leading to broader absorption bands. These bands can be even broader in solutions due to interactions between the solvent and solute. The Beer-Lambert law is an important relationship in absorption spectroscopy, including UV-Vis. It describes the absorbance of the sample substances in a solution. This law is represented as absorbance A due to the presence of a specific absorbance substance being equal to the product of three quantities. The first quantity, epsilon, is the molar absorption coefficient of that substance, also known under the obsolete name extinction coefficient. The second quantity is the concentration of the absorbing substance in the solution, and the third is the length that the light beam has traveled through the solution. Note that absorbance due to different molecules is additive, meaning the total absorbance of a mixture is the sum of the absorbances of its components. The molar absorption coefficient epsilon is a measure of how strongly a substance absorbs light at this specific wavelength. Its value depends on the nature of the molecule and the wavelength of the incident radiation. When analyzing an absorbance spectrum, it's common to measure the absorbance at the peak, which represents the maximum absorbance in a specific wavelength range. This gives a quick indication of the most prominent absorption feature. The peak absorbance is associated with the molar absorption coefficient at the specific peak wavelength. However, to get a more comprehensive understanding of the absorption behavior, it is possible to integrate the entire peak. This integral value represents the total absorbance over a range of wavelengths and provides a measure of the overall strength of the transition. The integrated absorbance relates to the integrated absorption coefficient across the entire peak.